Today we'll study about the poison apparatus. So the difference between the poisonous snakes and non-poisonous snakes is the presence or absence of poison apparatus. As all the poisonous snakes have poison apparatus present in their head, which is not found in the case of non-poisonous snake. The apparatus includes a pair of poison gland, their duct, fangs, and muscles. Now talking about the poison glands, two sac-like poison glands are situated, one on either inner side of the upper jaw, below the eyes and somewhat behind them. So this is the position of poison glands which is present in the poisonous snakes. They are basically considered to be the modified superior labial or parotid glands. In the case of sea snakes, the glands are small and oval in shape, whereas in the case of vipers, the glands are large and tubular. So this shape of gland is entirely dependent on the type of species. Each gland is thickly encapsulated with fibrous connective tissue and mostly covered by the fan-shaped constrictor muscles. These constrictor muscles are often known as temporal or masseter, and it is the stretching of these constrictor muscles during the biting which squeezes the poison from the gland into the duct. Here you can see in the diagram, these are the constrictor muscles, and stretching of these constrictor muscle during the biting squeezes the poison from glands into their respective ducts. So this was all about the poison gland. Now talking about the poison duct. So what are the poison ducts? Basically, a narrow poison duct arises anteriorly from each poison gland, which ends in the base of the poison fangs to enter its groove or canal. Here in the diagram, you can see these are the poison duct which are arising anteriorly from each poison gland to the base of the poison fang to enter its groove or canal. Next, talking about the fangs. Fangs are basically the specialized teeth which are attached to the maxillary bones. These are curved, sharp, long and pointed. Their function is to serve as hypodermic needle for injecting poison into the body of victim during the biting. Now when a functional fang is lost or damaged, it gets replaced by one of its reserved fangs. Here you can see in the diagram both the functional fangs and the reserved fangs. So on the basis of structure and position, three types of fangs are found in the different poisonous snakes. These are as follows. This type of fang is the solonoglyphous fangs, which is present in the case of rattlesnakes and vipers. In these snakes, a large functional fang occurs in front of each maxilla, whose base is covered on all sides by a sheath containing a few reserve and developing fangs. The main characteristic of these solonoglyphous types of fangs is that these are movable, and when the mouth is closed, these fangs turn inside to lie close to the roof of the mouth. The first type of fang is the solonoglyphous fangs, which is present in the case of rattlesnakes and vipers. In these snakes, a large functional fang occurs in front of each maxilla, whose base is covered on all sides by a sheath containing a few reserve and developing fangs. The main characteristic of these solonoglyphous types of fangs is that these are movable, and when the mouth is closed, these fangs turn inside to lie close to the roof of the mouth. So this was about the solonoglyphous fangs. Now the next are proteroglyphous fangs. which is found in the case of kraits, cobras, coral snakes, and sea snakes. In these types of snakes, the fangs are usually small, and again, these are at the front of maxilla, same as the case of selenoglyphus. But the difference is these proteroglyphus kinds of fangs are permanently erect, these are not movable, and these fangs are grooved all along its anterior face. The third is the opistoglyphus kind of fang, which is generally found in the family of Coluberidae. The fangs are small, but the difference is these fangs lie at the back of the maxilla instead in the front. These are found at the back of the maxilla, and each one is grooved along its posterior border. So these are the three types of fangs found in different kinds of species of snakes. Here you can see in the diagram the Solonoglyphus fangs. It's LS and TS. Talking about the snake venom. What is a snake venom? A snake venom is a poison which is secreted by certain poisonous glands, which we have already discussed, that is found in the head of the poisonous snakes and is injected into the body of the victim through the fangs that serves as hypodermic needles. Talking about the physical properties of snake venom, it is generally a clear sticky liquid which is of faint yellow or greenish color. It is tasteless, odorless, and acidic in nature. Basically, snake venom is a complex mixture of enzymes and some specific toxins, and it also serves as a good digestive juice. So, talking about the snake venom, it is only fatal when it is mixed in the blood. It can be swallowed, and if there is no scratch in the mouth or alimentary canal, it can be passed out with the feces without causing any harm to the individual. It can also be precipitated in the reagents such as silver nitrate and potassium permanganate, and it can be dissolved in the water and salt solution or glycerin and can be equally poisonous. Most of the snake venoms consist of both the neurotoxic and hemotoxic factors, but in general, the venom of vipers acts principally on the vascular system, therefore, it is hemotoxic, whereas the venom of cobras acts upon the nervous system, therefore, it is neurotoxic in nature.